So, continuing on, we're almost done with this topic on membranes. We're going to talk about what's called facilitated diffusion. If I was a facilitator, what do I do? What's facilitate mean? If I was to say, like, maybe there's an event and I had to facilitate transportation, what's that mean? Yeah, carrying it out, maybe be organizing it, making it happen. Okay? So facilitated diffusion is a special kind of diffusion that is facilitated by membrane proteins. Okay? So we should have this down by now. Okay? We've talked about over the last couple weeks how the shape of a molecule, the polarity of the molecule affects its ability to pass through the plasma membrane. So that's kind of like the ability to pass through the plasma membrane depends on size, big stuff doesn't fit through, shape, so certain molecules have a shape that doesn't work very well, polarity, because the phospholipid membrane is hydrophilic on the outside, hydrophobic on the inside. Things with polarity or ions don't want to go through it. And sometimes it can affect on where amino acids are on the surface of a protein. So certain amino acids will have polarity or shapes that are incompatible with going through the plasma membrane around the phospholipids, okay? So that's just a review. We should be at this point already. Let's go on to the next one. So, like we said, molecules can go through the membrane, but the ones that can't need to go through facilitated diffusion. Okay, so facilitated diffusion, like we said, it's something helping it through. And that's going to happen in what we call channel proteins or transport proteins. Okay, so these give a sort of a door, a pathway, a pipe, a tube, a pump for very specific molecules to pass through. So it's almost like if I had a big garage, I have a door for cars to pass through, and maybe I have a door for people to pass through. So I know that you can walk through a garage door, but you get the idea. Certain doors are for certain things, okay? So make sure we know what facilitated diffusion is. Make sure you could draw a protein channel and know that it's for very, very specific molecules. Let me get my face out. Okay, there's nothing back there. All right. So these channel proteins or transport proteins, they are required, okay? This is a must for facilitated diffusion. And again, the reason why molecules are too large or something about their chemical makeup doesn't allow them to get through the phospholipid bilayer. It has to go through here, okay? And like I said on the last slide, I'm saying it twice because it's important, okay? It's not like if, say you need to get sodium into your cells and some drug blocks all the sodium, okay? A lot of the lidocaines and novocaines will shut down a sodium-potassium pump in your nerves, and that's why you can't feel things when you're getting surgery or dental work done. It's not like other proteins, channel proteins or transport proteins will just let that happen. The doors are very, very, very selective, okay? So specialized is a big deal, okay? Specialized. They're not just going to let everything pass through. All right, one particular kind that we're going to look at is an aquaporin, okay? And I hear the words aqua, which means water, and pore, which is like a little hole, okay? So these ones are sort of specialized for water to go through, and they let a lot of water get through at once. And that happens because, as you know, water concentration levels in cells is a really big deal. If cells dry out, they'll shrivel up. If cells have too much water in them, they can burst open. So cells like to keep their water concentrations controlled. Who's going to make that happen? Aquaporins. They do it quickly, okay? They do it in large amounts. Can water go through here? Yes, but slowly. Yes, but uncontrolled. So this is slow. This is fast. All right. So one of the things we need to pay attention to here are their ions cannot get through here. So no ions go through here. Ions must go through channel or transport proteins, okay? So some of these can pump ions both ways or different ions at the same time, and biologists love that. We're going to show you one of those a little bit later. I think we like it just because it's so efficient. It pushes one thing out and another thing in, which sets up a polarity on the two sides of the membrane. So why doesn't this work? Well, ions have an affinity or an attraction. So let's say it's Na+. They kind of want to hold on right here, and they won't go into this hydrophobic area. So you have to use a special door to get them through. All right, here's a little bit more complex picture. This is something we talked about. When ions move across the membrane, a charge can build up. And it doesn't necessarily have to be positive on one side, negative on the other. It just has to be, when I say that, I mean ions, okay? You could have negatives on one side, negatives on the other, but those charges can be different. So you could have like a higher negative amount on, say, this side. So this would be more negative. Each one of these negatives would represent like a negative ion. Or you could have fewer negatives on that side. It will still set up a charge because there's more negatives on this side than there are on that side. So a charge difference is what we're looking for right here, okay? So when that happens, the cell is polarized, which just means one side's different than the other. Usually, the insides of cells are negative. So let's jot that down somewhere, okay? Inside of cells tends to be negative. Now, it can temporarily change, and it does, and that's how your nerves work, okay? We're going to get into that, but not in much detail right now. So, this takes a lot of energy to pump these ions onto the proper side. Compared to every other action in your body, ATP being used in muscles, digestion, keeping your heart going, keeping digestion going, all these metabolic processes, I have a question for you. What percent of ATP, or you could look at it as calories, are burnt in your brain? Say that louder. Well, give me like more or less than what percent? Just give me a guess. 50, around 50% are burnt in your brain. Now, if you have a bigger body, it takes more ATP to get you going. When I walk up the steps, I'm a really big, heavy dude. I'm going to use a lot more ATP than like some 90-pounder, okay? So it depends on a lot of factors, but it's around 50%. And that includes like when you're at rest. What do they tell people with concussions they need to do? Cognitive rest, okay? You can rest your brain, but it still is going to use huge amounts of calories, okay? And it's because, always come back to the because, pumping all these ions is what makes nerves work. And so constantly resetting the polarity here, every time a nerve fires, monstrous amount of energy used, okay? So 50% would be like your nerve, let's expand brain out to nervous system, because that includes like your spinal cord and all your major nerves. So that's a cool trivia fact, but don't get lost in the sauce. Why? Because active transport happens in nerves constantly, and it has to happen quick, okay? So imagine just tapping your foot. That's a nerve signal to go down, reset, a nerve signal to go up, reset. That nerve, it goes from your brain, spinal cord, all the way down to your foot. So that exchange of ions is happening along that whole nerve. So just tapping your foot is going to use a lot of energy. Now, don't think that you're going to be, well, I'm on the tap your foot diet. It's not going to be that much. Or I'm just going to think really hard and that will help me burn extra calories. It does, but it's not enough to like take that 50% up to 80. Okay, that's kind of like a fixed level. When you're asleep, it goes down. When you're just kind of zoning out, it goes down. It does go up by a few percent and down by a few percent. Not enough to make you like, I'm just going to lay in bed and think and I'll lose weight. No, okay? Especially if you're eating ice cream while you do it. All right. So these channel proteins are required for active transport. Now, not all forms of active transport, endo and exocytosis are active transport, but this takes a lot of energy. And that's kind of what I just spent a lot of time talking about. This moves things from low to high. And it can do it fairly quickly in certain cellular situations, okay, metabolic situations. Nerve cells are the big one. So let's kind of look at that briefly. This is a review. You've seen this slide before, okay? 3 major types of membrane transport. Diffusion through the phospholipid bilayer. Passive transport through a channel protein. Today we're talking about active transport. So active transport is facilitated diffusion. They're the same thing. The big one there, energy, and sometimes a lot of it. Okay. So these protein pumps often look like, I don't know, little pills or M&Ms or something, and they will have some sort of pathway through them, and ATP comes in, and what is often called phosphorylizes the protein. That's going to change its shape And then usually the phosphate jumps off and it changes its state again, and it ejects it on one side of the membrane or the other. So there's a lot going on here, okay? If you were going to take marbles and put them from one bucket to the other, you have to grab the marble, move it to the other bucket, and let go of the marble. So for a protein to do this, it's kind of really chemically cool. The ATP comes in, phosphorylizes the protein, and that makes, in this case, hydrogen want to bond to it. Then it opens to the other side of the membrane, and it has to make hydrogen not want to bond to it. So changing how chemicals bond very quickly is what proteins, in this case, are doing. How does that happen? Usually the protein will change its shape, and that will make it attractive or unattractive. Just like my hand changes its shape, move it over, changes its shape. So this is, on a biochemical level, you have to stop and appreciate this, okay? Usually when chemicals bond in your chemistry labs where you're using metals and inorganics, or a color change, it happens, and reversing it in most cases is not very easy. Like if I burn magnesium and make magnesium oxide, it's not easy to make the oxygen let go of the magnesium and go back to a metal, okay? If you let iron rust, not easy to make it unrust, okay? So the fact that these are bonding, unbonding so fast is really, really cool. If this happens slower, life forms interacting with the environment might be at a level that was so slow they couldn't maintain homeostasis. So biological reactions have to happen at a rate that allows homeostasis, okay? So remember, I say the same thing a lot because it's important. Each particular pump matches up with a specific ion. In this case, it's hydrogen. Okay, we're going to study outside of this lecture I'm not going to go through this right now, okay? A really, really cool pump called the sodium-potassium pump. And why we love this, every ATP pumps 3 sodium ions out and 2 potassium ions in. And the pump is going in different directions, multiple ions, and all using one ATP. It's really hard to show without an animation, and I didn't want to try to squeeze an animation on the screen during the lecture. So outside of this lecture, I have an ed puzzle for you to watch on how that happens. And biologists love this because take a look at this picture. We see potassium going out, sodium going in, 2 potassiums in, 3 sodiums out. This all takes 1 ATP. This pump kind of works like this. It'll open up and it will load the sodium ions in. Boop, boop, boop. 1, 2, 3. I don't have anything to use for a sodium ion. I can't hold 3 things at a time. So picture there's 3 marbles in my hand that represent sodium. They stick to the pump. The pump reverses like this. The 3 sodiums come out, boom, 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 out. And remember, have to be attractive to sodium, have to break that bond with sodium. So that happens really quick, okay? So the 3 sodiums just came out. 2 potassiums now hook up here, and the pump resets. The potassiums break off and go into the cell, and that's all happening right over here. It's just, you just can't appreciate it until you see an animation of it, so that's where we're headed next. All right, where does this happen? We already talked about this. quite a bit with how much energy your brain uses. So nerves are made up of cells called neurons. So is your brain. These carry signals throughout the body very, very quickly. And they do that by changing their polarity. So they'll have passive and active transport channels on them. So nerves are really good to study, to learn this stuff. And using huge amounts of energy because they're pumping so actively. This is sort of a step-by-step version of this. You can see the three sodium ions loading. They're orange, and then they get ejected. The potassiums come in, and they get ejected on the inside. We've got some ATP making this happen. It does that by breaking off a phosphate, and you can see ADP here at the end. So, what does this do for your body? Makes it negative on the inside of your nerve cells, positive on the outside. When that changes, it kind of initiates like a wave in the stadium where all those ions sort of get that wave going. Then for the nerve to reset or repolarize, it has to pump that the other way. Okay? So, Tori, we just ended, so that's perfect timing. All right, so after this lecture, you should be able to do these things, okay? You should be able to tell me, what's facilitated diffusion? Well, Mr. Sater, that's a certain kind of diffusion where a protein makes very specific ions pass through the membrane, okay? You should tell me what the role of these membranes is when we talk about this. Well, Mr. Sater, there's a protein for every ion or every substance, and they are pumps that use energy. And what kind of energy does it use? Well, Mr. Sater, you use ATP, and as it gets used, it turns into ADP. All right? And you want to outline how one of these works. Well, that's going to be a little bit outside the role, the scope of this lecture, so we'll have a separate assignment to address that. All right. So this was 2.7, facilitated diffusion. We're really going to try to get through this unit here in the next couple days. So look for a few more of these and we'll be done.